start, and then people will jump in whenever they are present. So welcome to this morning's Centers of Excellence in Hematology Microscopy Monday. I hope you'll share this information about this great opportunity to learn good microscopic techniques, cutting edge methods that are being used, and sponsored by the Centers of Excellence in Hematology. And we have two great speakers today, which will give them an opportunity to sort of introduce themselves in a minute. I'm going to share my screen for a minute to just tell you about the Centers of Excellence in Hematology. And this is our website. If you go to it, it's really simple, cceh.io. And on this site, you can find all kinds of activities, upcoming events. Here's our Microscopy Monday event. What I want to draw your attention to today is funding opportunities. So I'm going to click on this link, and this one always pops up on me. If you scroll down this page just a little bit, you'll find funding opportunities coming up. And type A applications are $5,000 to $12,000 worth of credit at any of the cores that we sponsor. These are available year-round. You can apply for them. They're a very simple application. And the type B applications are called for about twice to three times a year. We have a new application deadline, which is February 28th. These proposals are up to $75,000. And you can read the details here. There's also monies available for travel to train at a core if you need to do that. And I highly encourage you to go look at this opportunity if you're thinking about a new direction for your research, or you have a brand new lab and you need some support funds to do research in non-malignant hematology. And now I'm going to stop sharing. If you have any questions on that, you can feel free to reach out to me. My email is right here, and I'm happy to help with anything on that. Okay, and so without further ado, we're going to let Dr. Guo go ahead and share her screen.
Let's see. Share screen.
While she's doing that, I just want to invite anybody who's going to have questions to go ahead and interrupt. That's been agreed to. If you want to populate a question in the chat, that's also okay. I will invite you to ask a question, and let's go from there. Thanks again for presenting.
Okay. Do you see my slides?
Yes. Yes.
Good. All right.
And if you're not speaking, would you please mute yourselves and then just unmute yourself when you do have a question?
Okay. Well, again, this is a great opportunity for me to really speak on behalf of Yale CCEH, particularly the imaging core. So today you see we have two speakers. The way I thought to structure today's presentation is for me to take the first 30 to 35 minutes to give an overview and some general examples of this topic, imaging, live imaging of hematopoietic cells. And then the next 15 or so minutes will be presented by Vanessa Scanlon. I'm very proud that this is it's a live and breathing example using this live cell imaging platform to really push the front of important questions in the hematopoietic stem and progenitor field. Okay, as Diane said already, please interrupt me. And let's get started. So the first question is, why do we bother with live cell imaging? What is so cool about it? Why do we want to push forward? So I really stole a bunch of pictures or figures from fabulous reviews by Tim Schroeder. You probably all know that Tim and his team is one of the really pioneering leading experts in utilizing long-term live cell imaging, particularly for hematopoietic stem and progenitors, to answer important questions over the years. So the first point I want to make, this is also taking from his paper, a review, is that live cell imaging data inherently contains single cell data. So nowadays we hear all the single cell multi-omics, spatial omics. But what's not really appreciated to the full extent is that whenever you take videos of the cell type of your choice, they're always presenting to you as a single cell. Each cell does their own different things, so you get different sort of behaviors for all the lineages. So in this particular figure, Tim plotted that there are two axes. The x-axis is time, temporal resolution. The y-axis is about the spatial resolution, whether single cell or, he's using the blender or grinding up everything to take a population average. And you can see with the live cell imaging basically is occupying a high sort of coordinate that having both really unique temporal and spatial resolution, for which even the most updated the single cell omics data would not give you the time resolved information unless computationally you can infer for that. The other thing is the different lineage trajectories tell you different stories. For example, this is taken from Tim's another review. You can see the reference here is with a very simple lineage. You begin with 4 white cells. After a period of time, we can end up with 8 cells, of which 4 of them remain white, 4 of them are red. And what happened during this black box could be a number of any of this. If we just know the beginning and the end, we would not know what happened in the middle. So what happened in the black box. And each of these black boxes tell you a different story, implies a different mechanism, gives you a different hypothesis. This basically is why the live cell imaging approach, to watch the 4 white cells to gradually over time become the 8 cells, is going to give you the kind of hypothesis otherwise would be impossible to have. These 2 reviews I just referred to are here. You're welcome to read in more detail, and Tim summarizes very nicely. So why live cell imaging? I'm going to dwell a little bit more on that to give you more substance. Why is because there's a great extent of cellular heterogeneity. We know that. And let me back up a little bit. Okay, so for a lot of the biologies that we're interested, at least particularly for me, I am interested in, this comes from a rare cell type. So usually 1 in every 1,000 or 1 in every 10,000 cells, for example, hematopoietic stem cells, or the particular multipotent progenitors that you're studying. They're rare. I mean, we can prospectively enrich and purify them. But even that, it's probably we're down to 1 in every 3 cells, 1 in every 2 cells. With this kind of heterogeneity or low frequency, so you really need to interrogate those relevant cells one by one to get your biology. And usually, so I like to also say, for example, the drug resistant cells or metastatic or cancer stem cells, probably not particularly relevant to the non-heme topic, but these are also great examples for rare cell biology that requires the kind of interrogating one cell at a time. And the dynamics. My lab particularly has been spending a lot of time trying to take apart the cell cycle dynamics. And their cell cycle dynamics could be the cell cycle phase. We know G1S, G2, and what happens in distinct cell cycles, there could be difference. But there's also signal transduction. And Tim Schroeder, for example, in this case, they imaged quite a lot. Sorry, this is a typo. I should say this is ERK, ERK, MAP kinase ERK. calcium or NF-kappa B or a number of your favorite, could be your favorite transcription factors, they don't just stay in the nucleus or sit on the chromatin to transcribe. They actually translocate between the cytoplasm and nucleus, go in and out. And all these are classic examples that are flashing or oscillating, comes in waves. And the sort of periodicity varies between from some waves are on the seconds time scale. Others could be on hours, or certain other things could be on days, or circadian cycle, right? That by definition, we know it comes every 24 hours. How do we do that? And cytoskeletal remodeling. So all these are incredibly dynamic and beautiful to watch. But if we're only looking at one snapshot of the biology, it could be very confusing. In subcellular compartmentalization, for example, the shuttling I just referred to for the transcription factors or cytoskeletal remodeling, it could also be very oscillatory. Cellular dynamics. We usually we're used to seeing the for example immunofluorescent images for a particular snapshot because we fix the cells. But whenever you look at the live cell movie, depending on how sort of frequent those images are taken, it's incredible that how much cell interaction, the cell coming together with each other, forming synapses, or taking a piece of the cells off the other, trogocytosis, or migration, or one cell crawling through the other, or one cell crawling underneath or coming back from the other side. All this incredible biology would not, we're not imaginary enough until we have seen this. So I also want to make a disclaimer is that I'm not trained a microscopist, not like my colleague Jorg, for example. I really started playing with this live imaging, or sort of learning through doing or making a lot of errors. So the way I'm presenting this is really from the operational or biologically minded. So the challenges to overcome before you can image your favorite cell type. First of all, is that you need to have a microscope with environmental control that lets you continue to cultivate or observe the biology without your cells dying on you or behaving weird. So that's the minimum, right? You need to have temperature control, the humidity control, CO2 control, just like any incubator. So suppose that is checked off. Then the other things we need to consider is, to image them, we need to shine light on them quite extensively and repetitively, sometimes it's continuously. And this generates unavoidable phototoxicity. And this is not talked enough, but until you run some pilot experiments, you will see that the imaged area, the cells in the imaged area compared to the cells that are right outside of the imaged area, they're different. Sometimes you see literally a hole in the imaged plate because the light is basically burned through everything. So then the ways to reduce phototoxicity is something to be considered. And what I'm not listing here also is the autofluorescence, background fluorescence, because a lot of the media that we use contains serum, contains phenol red, contains riboflavin. All those things give rise to autofluorescence. And if your signal is weak, those pretty much takes away your chance to get a crisp image. And the resolution, this is both temporal and spatial. Temporally speaking, is what kind of biology we're talking about. If we're just watching, for example, cell migration or cell-cell interaction, then at the whole cell level, it's enough. But if you want to see nuclear cytoplasmic shuttling, then you probably need better, more advanced. And I meant spatial. Okay, so temporal is for time, then how quickly your biology is happening, from seconds to hours to days. For the imaging that we're talking about could also be, most of the imaging that would be sort of informative requires a certain kind of fluorescent signals. So where would the fluorescent signal come from? Could coming from in a variety of approaches. One is antibody staining. I believe Vanessa will give you some examples, but it's quite often used. That is the easiest if you can find a good antibody and good antibody staining protocol. And fluorescence reporters and probes that need some literature search, or you're engineering the mouse genome yourself, or human cells. And also we would like the biology of interest to be validatable by other means, because if the only way that you're addressing your question is by imaging, we really have no ground truth to compare it to. The last one, I think, is the hematologist, or this group would be mostly sort of be affected, but not covered by most of the imaging sort of tutorials. So how do you hold yourself still? Because this is not a small task, and we'll see more examples coming up. As I said, I'm not a trained microscopist. So my experience started on this machine, which is part of the Yale CCEH core, the VivaView subunit. What you see here, this is the microscope. Underneath of it, you see a sort of rotating plate capable of holding 8 simultaneously, 8 35 millimeter dishes. And this is a self-enclosed sort of incubator microscope inside this incubator. So the cell, even over days or weeks of culture or imaging, it won't be affected. So my experience really started on here, and later on, my lab acquired another sort of self-enclosed high throughput imaging sort of microscope or unit called Micro Express, Image Express Micro, from molecular devices. And I think Vanessa again has access to one of the even higher upgrade of those. So I don't think the stuff we're talking about is specific to any particular imaging unit. As long as the imaging unit or microscope can give you the environmental control over a long time, what we're talking about should still apply. But if any one of you want to try the any of the imaging instrument that we have here, I think our imaging core is equipped or is part of our goal to help you to try that. And we just heard from Diane the sort of support. If you want to try yourselves on our microscope, you can apply for the funding to come here. We would love to help you to try that. Okay, some of the key considerations. Where does the biology take place? In vitro, ex vivo, or intravital? The intravital one, of course, it is the most complicated or involving one. It requires the live mice under anesthesia to be. But supposedly that some aspect of biology can be recapitulated by in vitro or ex vivo culture, and this is the sort of the condition we're talking about. How fast? In the sort of temporal resolution part, I talked about that different. How frequent? Does every cell in your imaging dish show the biology, or only a few? That determines how many cells you need to image. If every cell does this, you only need to image, for example, 5 cells. If 1 in every 100 cells gives you that biology, you probably need at least 500 cells. That's what I'm talking about. How big is the resolution? Again, are we talking about a part of the membrane that does certain things, like the asymmetrical distribution of something on the hematopoietic stem cells? Then we really need to zoom in on one tip or one side of the cells, or really the whole cell would be fine. How steady? This again is the blood cells do weird things. They are not only migrate, they can float, they can rotate, they can stretch, they can really burrow through. It's all these crazy things they do, and how do we deal with them? signal varieties, and the informative ones could be all this visible light or fluorescence. And I'm not going to cover the luminescence because none of our instruments currently can deal with this, but it's just for FYI. All right, even for fluorescence signals, we have many varieties. There's constitutively fluorescent, genetically encoded tags, or fluorescent binders. The antibodies is one of them, and they're genetically encoded fluorescent antibodies. We'll see some examples. And fluorescent chemical probes for example, calcium. Conditionally fluorescent, there's a photo conversion, photo bleaching, or photo, you can convert them, turn on the fluorescence conditionally. And I'll also show you one of our sort of probe as a timer protein. And these would be FRAP or FRET signal or imaging would be more toward the dynamics that I will talk later. So this is one example. This is not from our work, but one of these sort of more or less now classics now, to really visualize how hematopoietic cells were born, quote unquote, from the pluripotent ES cells in culture. This is one of the movies. The paper that described these movies are here. You can find it. These are layers of mouse embryonic stem cells, and you can see them giving rise to giving birth to hematopoietic cells. This is a movie acquired during 4.5 days at 4 minutes intervals. You can see that that when these originally the cells were all sort of layer sheet, and at the end you can see they acquire the typical hematopoietic morphology, and then you can annotate, construct the lineages. This is another classic example to actually visualize hematopoietic cells being born in the mouse aorta. This is accomplished by the one is the GFP transgene knocked into the SCA1 locus. The other one is endothelial marker stained by the CD31 antibody. I recently switched computers, so unfortunately this movie does not play on my computer right now. But you can find the original published movie at this paper. Okay, so fluorescent binders antibodies. Vanessa is going to show you more, and these are some really old papers, but it shows you this. For example, if your cell type or biology can be detected by a cell surface antibody, and then you can perform the imaging, and at the end either fix or live cell to identify the cells that you imaged through these antibodies. There are protocols published, and Vanessa will show you that one of the kind of, or several type of antibody that she figured out how to do that. The limitation is that most of these antibodies are limited to cell surface proteins. And if you want to image the sort of longer duration after you're staining it, it could be complicated by the antibody internalization, or whether the antibody staining itself changes the cell biology. So temporal precision could become limited as well after this. But this is something to consider. I'm just putting it out here for fun. You must have all heard about the bow or the colorful mouse proteins. And for this audience, we know that, for example, the Scanlon lab later published the bone bows or the rainbows, the brainbows, if you cross it with the specific type of Cree, that would give you different cells. But I don't think I've seen the sort of long-term imaging using such a sort of identity reporter in this kind of high complexity. I mean, I think if there's sufficient justification to do this, this should be giving us some really unique and beautiful movies. So this is another type of fluorescence, which is cell identity reporter, meaning that what the color of the cells would tell us what they are. Then what can be imaged? The other type of fluorescent signal, of course, is the fusion proteins. This is, again, one of the very ancient sort of a collection of reporter mouse. Essentially, all the subcellular organelles, they've tagged it, light it up. We can light it up with these fluorescent proteins. And recently, of course, the Allen Institute has been pushing forward or contributing to large collections of endogenously fluorescently tagged human cells. So if there's something of the interest that you want to see, and these would all be excellent sources. The other kind of fluorescence signals could come from fluorescent binders. The examples here I'm giving are called chromobodies. What are these? These are single chain camelid-based antibody. It's not unlike our Y-shaped heavy or like multiple chains. These chromobodies is basically just one chain, so it's easy to be encoded in a plasmid. And then if you put the GFP or cherry or fluorescent protein into this single chain antibody, then you generate a genetically encoded fluorescent tags to the antibody. So for example, this we could express the GFP chromobody against the actin into the cells, then you could visualize the actin dynamics in live cells. Why am I putting it here is because the Yale's imaging core has acquired 6 chromobodies. I'm only listing 5 here. I don't know why only 5. But for example, these 6 antibodies chromobodies we acquired can visualize actin, or I know actin or nuclear actin specifically, or cell cycle. This one of them I think is the PCNA, proliferating cell nuclear antigen, or nuclear envelope. Why I'm making this point is because each of these chromobody construct is costing that at the time I think is something around $800 or $1,000. So we basically bought them all together, and also we subcloned these from the plasmid form into lentiviral form, so which is much more convenient to use into hematopoietic cells. They don't like to be transfected, for example. So this resource has not been sufficiently utilized, and my lab, for example, we have been using quite a bit of the nuclear actin chromobody, and those images are quite interesting. Okay, here I think this one can play is the actin dynamics. This is not by actin chromobody, but another live actin binder, and this is the kind of dynamics you can see from a movie, from a single cell. The hematopoietic cells won't be usually are not going to be this spread out for you to see, but again, if there is a specific interest, subcellularly, the whole cell or in specific organelles, there should be probes for us to see that. So what it really comes down to is, even if not everything is ready, meaning the probes or signals are not all there ready for you to start the experiment yet, it only whether you can see the biology is coming down to the question, what would you like to see, or what's the question you would like to attack? Here I'm just going to go through some quick examples for the questions that my lab specifically has been interested in. One of which is, we know that blood cells, some of them at least, can become cancer, and we really want to see the cancer initiation process from when the cell is a normal hematopoietic progenitor to become a leukemia. In this case, of course, as I said, then we need to do some preparation, right? So we have the scope, then we need to make sure we have the kind of fluorescence we are looking for. So in this case, we acquired the MIS1 GFP, which is a back transgenic, and cross it with our oncogene inducible mouse. And this oncogene we created which is a hematopoietic oncogene knocked into the HPRT locus and crossed with the ROSA 26. So in mice carrying both of these alleles, we know all the GMPs essentially, once you turn on the doxycycline, they become transformed into, or they can be transformed into leukemic blasts. So then we started imaging this process. These are all GMPs. They're sort of began as the same cell type, but you can see at this time, I don't know whether you can see that. This is one of the MIS1 GMP reporter positive cells, which means it should have transformed already. And what I want to draw your attention is until we see this movie, we would never be able to appreciate how migratory these crazy cells are. Let's see. In the beginning, they're pretty same looking. And here comes this migratory crazy cells, and that's a transformer. So we haven't really followed up on our, the leukemia or cell of origin for initiation. Are they more migratory? Is the migratory machinery contributing to any of the biology? That's something we're following up on, but we don't know what the answer is. So what I alluded to is that what's not talked about is how to hold still of the hematopoietic cells that you want to image. So to address this question, again, different cell types requires different tricks. So for example, what we did here is that we did micro mold that essentially this whole green piece is like a quarter coin size. And in the center of it, it's essentially a mold that you can cast essentially a tiny 96 well culture device into this mold. And what this allows us to do is that we can plate single cells into these wells, and there is a sort of a zoom in one cell, and because it's too zoomed out, you cannot see these little cells dividing. So basically the trick here for us to image this process is to confine each cell, not really confine them, but at least their migratory territory is being confined to these tiny wells. So at the end of 7 days, we can see some of the individual cells has turned into a transformed colony. Some of them sort of give rise to this not remarkable, perhaps differentiated progeny. The other biology my lab has been interested is in cell cycle. And if there are people who are interested in the cell cycle phase progression, this is a pretty classic movie for the cells to change fluorescence based on their cell cycle stage, expressing the cell cycle phase reporter, what nowadays are known as Fuji reporters. And these are available now both as lentiviral constructs or mice, transgenic or knock-in. So what we have done in the past is not really particularly concerned about the cell cycle phase, or we are interested in that too, but we have been interested in the whole cell cycle length from one mitosis to the next mitosis. How long in terms of hours? Is the 8-hour cell cycle or 24-hour cell cycle? What we did is to fuse a color changing protein called timer with a H2B histone. So you can see, or the timer protein, what they are is when they're newly synthesized, they're blue proteins, and this version, I think, give them 2 hours, they'll turn permanently into red. Let me play this again. So you can see at this time, these two cells have synthesized the new fluorescent timer protein attached to or fused to the H2B, but the red fluorescence is just about to come up. And then later on, all of them sort of change, and the red protein stays, and the blue protein keeps on being made. So from this kind of dynamics, we know that the faster cycling cells would be bluer, and we can validate that because this is whatever in the transgenic or the knock-in mouse tissue sections, we can see the proliferative cells usually validated by the EDO pulse label, coincides with the blue cell zone. So this is how we know. Oh, okay. So it can also be measured by flow cytometry. For example, the BAF3 cells here, when we feed them in the complete IL-3 condition, most of the cells grow happily, they're blue. And if we starve them for the one cytokine they absolutely require, IL-3, you can see the whole population shifted toward the red. And this reporter, again, has been made into a mouse, and we can cross them with the leukemic mouse. Now we can see, of course, the leukemia cells are blue shifted on this axis. And I know that this tool or reporter mouse has been very welcomed by, at least by the red cell progenitor community, and quite a few labs have started using it. This is my last slide. Is that in addition to these biologies, if we change a little bit of the imaging modality, the live cell imaging would allow you to ask for molecular kinetics. Because a lot of us, when we're interested in the sort of epigenetics or chromatin changes, one of the things to answer is how does this chromatin landscape change, right? So, and when I say that, I don't mean by which chromatin is open or which part of the region is open. So what I meant is, for example, we've made a pair of fusion proteins. Again, that's mouse models. For example, in this one is H1.0 GFP fusion mouse, and each cell in these mouse would be having a green nucleus. And if we use high power light to sort of poke a hole, bleach that spot, and by watching how quickly each cell recovers, it informs how dynamic the chromatin state is. We're still using these mice to do things, but if this is something that could be of interest to your cell type, I'm more than happy to share the mice even before we publish them. I think, yes, my time is up. I would, pass the where is it? Oh, okay, stop share. Yes. So I think next we'll see some really beautiful movies, long-term movies, lineage annotated from Vanessa, who I'll let her introduce herself.
All right, thank you so much, Shangqin. Can you confirm that you see my slide okay? Great, fantastic. So thank you so much for this opportunity. It's really such a pleasure to be invited to speak about our work as an example of what we can do with long-term time-lapse imaging. I think in the field right now, we're sort of at this like really exciting moment of finally being able to couple really specific pieces of information that deal with cell phenotype along with cell function. But there's been a lot of challenges in terms of generating that data and being able to interpret it, for example, using cellular barcoding or transcriptional analysis, things like that. And so what I love about the long-term live imaging is the ability to actually couple these pieces of information together in real time to better understand the fundamental biology happening. So I'm going to kind of, for the sake of time, go kind of quickly, but please do feel free to interrupt with questions, or otherwise I'm also happy to answer questions afterwards as well. But when I started as a postdoc, I was in Diane Krause's lab at Yale in the CCEH, and they had just finished a study figuring out a way to isolate a particular progenitor called the megakaryocytic erythroid progenitor, the MEP. And so they had developed a colony forming unit assay that facilitated the growth of both the MEP as well as its daughter cells, which could be MEG committed or erythroid committed. And so in these colony forming unit assays, you very sparsely plate your isolated cells, like literally about 100 cells per one well of a 6-well dish. And it's in a semi-solid media with all of the nutrients and cytokines required for survival and differentiation of these disparate lineages. And at the end of the culture period, about 14 days later, you can assess the colonies that have formed, and you can look at the types of daughter cells that are present within those colonies. And so there are 3 types of colonies that we routinely see when we plate MEP. There are colonies that only contain megakaryocytic cells, colonies that only contain erythroid cells, and colonies that contain both megakaryocytic and erythroid daughter cells. And so at the time that this was published, it was great because it really demonstrated that there was now an immunophenotype that you could use to isolate the cell with really high efficiency. So out of 100 cells plated, about 70 of the cells are making colonies, and almost all of those colonies are containing the daughter cells you would expect. So I joined the lab as a postdoc, and I see this. And my very first question to my postdoc mentor, Diane, was, well, how come if these are truly bipotent cells capable of giving rise to both lineages, how come sometimes you get colonies with just one lineage? Wouldn't like a bipotent population always give rise to mixed colonies that have both lineages? And is that coming from contaminating lineage, like unipotent lineage progenitors that are just contaminating the MEP gate, this immunophenotype? Or does it actually come down to a question of probability, that just because a cell is bipotent and has the choice to make 2 lineages, is there a probability that it's going to sometimes just make one or the other? And so I started to think about ways to figure this out. And I thought, well, perhaps the cells themselves behave very differently. Perhaps MEP have a very different behavioral phenotype than a MEG-committed progenitor or an erythroid-committed progenitor. And how would I start to assess that? And so that's kind of where my introduction to long-term time-lapse imaging came from. And so because I was in Diane's lab, I also used the Olympus VivaView. Thank you, CCEH, for supporting that piece of equipment. I will say that now in my faculty appointment at the University of Connecticut, I also do these movies with a molecular device, Image Express Microconfocal. So it has the ability to visualize 7 different colors simultaneously. It has confocal ability, and obviously also retains the environmental controls necessary for keeping the cells alive for the duration of our imaging. But basically, I set this assay up just like we would set up the CFU assays. So I plated very few MEP in a dish, and in that semi-solid media. I'll tell you my trick for stabilizing the cells, because they are non-adherent cells, and it is a semi-solid media, but we want to be able to visualize the cells in focus for the duration of the experiment. What I do is I plate the cells in a very, very low volume, and I sandwich a second cover slip on top to minimize the range of depth that the cells can travel in the Z-axis. And so then I image, over the course of 7 days. I will say for the phototoxicity issue, we did need to adjust our image acquisition. So in the initial 24 hours that the cells are being imaged, we're only imaging about once every 2 hours. So we're reducing the frequency of imaging to try to reduce that the amount of light that's being shined on the cells. But we did also notice that after that initial sort of warm-up period, it's almost like the cells sort of developed a tolerance for the light. And so then we were able to increase our imaging frequency to every 10 minutes. And that was important because we were trying to capture individual cells as they were dividing, and then also keep track of those daughter cells and their daughter cells. So we didn't want to miss any divisions, and we wanted to be able to, with confidence, link cells between timeframes. And so that's kind of how we developed our image frequency there. And then at the end of the movie, because these are, I should have said, this is all with primary human cells. So these are either from mobilized peripheral blood products or from human bone marrow. We can do it from both, but we take these cells. They obviously don't have fluorescent reporters genetically encoded because humans aren't born that way. And so we use an in situ staining approach with some fluorescent antibodies, and we add them to our dishes towards the end of our acquisition period. And these antibodies sort of intercalate down through the semi-solid media and bind whatever cells are expressing the lineage marker that we're looking for. For MEGs, we look for CD41, and for erythroid cells, we can use glycophorin A, or alternatively, a slightly earlier marker is a drastic upregulation of CD71 in the erythroid committed cells. So we purposely add these antibodies at the end because we have, in our experience, tested and found that the earlier presence of these antibodies can also change the biology of the cells a bit. And because we're really trying to look at a natural process, as close to a natural process as possible, this is why we wait to add our fluorescent antibodies until the end. So then what we do is generate lineage trees from these time-lapse images where we watch a single cell forming an entire colony worth of daughter cells. And so we track the cells, and I could have a very long conversation with everyone about the analytical tools that are available to us when we're trying to analyze time-lapse imaging. But for the sake of time, I'll just say we are utilizing the Baxter algorithm, which was generated by Helen Blau's lab at Stanford University. And it allows us to sequentially follow the cells throughout the duration of the movie. And so we establish our lineage trees. And then, as I said, we only have fluorescence at the end. So at the end, we will false color our lineage tree based on that fluorescence. So we can identify which daughter cells are megakaryocytic and which daughter cells are erythroid, based on their fluorescence. And then we start to basically rewind the movie and work our way back up the lineage tree. And we false color those parental cells based on what's downstream of them. So if a parental cell has only erythroid daughter cells below it, then we false color that parental cell an erythroid destined cell, meaning that this progenitor is only going to give rise to erythroid daughter cells. Conversely, if all of the daughter cells are megakaryocytic, then the common parental cell we would call a megakaryocytic destined progenitor. And then working our way back up the tree, of course, because we started with a single cell that immunophenotypically told us that it was bipotent, what we look for is progenitors that share daughter cells of disparate lineages. And so as we work our way back up, we start to identify these cells that are upstream of both erythroid destined and megakaryocytic destined cells. And so then we call these bipotent cells because for sure they're giving rise to two lineages. There is a caveat to this kind of analysis that I want to share very openly, and that is that our confidence of our false coloring of the trees varies depending on the cells that we're looking at. We are very confident at the end of the progeny and what lineages they belong to because they are fluorescing with an antibody that has labeled them. But our confidence about what is already erythroid committed or megakaryocytic committed has a limit, because it's also possible, as I said before, that a progenitor although it has the potential to do two things, may just choose to do one. And that's why we're very specific in calling these intermediate progenitors destined, erythroid destined or megakaryocytic destined. But when it comes to the cells that we false color as blue bipotent cells, we have a very high confidence about those cells because for sure we can watch the movie and show that that cell is upstream of two lineages. And so I just wanted to share that caveat. And so here is a representative movie that we make. So that's a single sorted human MEP, and we're following it over the course of 7 days as it divides and moves around. Yes, hematopoietic cells do move in semi-solid media, but it's not a sort of floating away motion. It's an intentional motility that the cells are exhibiting. And so here I've already gone ahead and false colored the lineage tree for our convenience. And again, everything that's marked as red is cells with only erythroid progeny. Cells marked in green are cells with only megakaryocytic progeny downstream. And cells marked in blue are in fact bipotent with both lineages below them.
Vanessa?
Yes.
Can I ask a question?
Yes, please.
A couple of questions. First, I mean, with respect to are the cells ever at a density where, because you're tracking them, they all look alike, and you're just tracking them with the tracking software. So do you ever have an ambiguity in identifying a cell as, two cells go to one spot and then two cells come out of one spot? Who's who? Does that come up ever, or they're not?
Oh,
that
sure.
dense?
Sure. Yeah, so that's part of the reason why we use that second cover slip on top to make sure that the cells are growing in 2D so that we can actually be able to visualize cells as they move. There is still some transposition of cells, and cells can certainly switch spots in the
But
movie.
you can distinguish them.
Yeah, and that's why it's so important to image at least in this cell population, at least every 10 minutes, because the cells are moving, but not quite so fast that within 10 minutes you wouldn't be able to determine which cell is which in the following frame.
So what is the time frame of this whole movie?
So the whole movie is 7 days long. The first portion of the movie, so maybe up until about the 4-cell stage, is being done every 2 hours. But after that point, we're imaging every 10 minutes.
And then the last question,
Sure.
this cover slip you put over the top of everything,
Yeah.
are you concerned about gas exchange? Because basically you're depending on diffusion to come in from the sides of the cover slip now. Is there any,
That's right. Mhm.
is that an issue?
No, so at the beginning, we just kind of thought like, well, let's just see if this works. And we were actually pleasantly surprised to see that passive diffusion is fine, right? The cover slip, if you could sort of imagine it as a sandwich, a cover slip on the bottom, which is the bottom of the dish. And then, our cells in a very small volume in the second cover slip on top, there's a bit of a sandwich, but the edges are open. So that passive diffusion allows for not only nutrient transfer, but even for antibody diffusion as well, right? Because we're adding that antibody at the end and it's making its way all the way down under the cover slip and reaching the cells so that they can fluoresce.
Thanks.
So yeah, but it was a really nice, like I said, pleasantly surprised to see that approach worked. But okay, so we can derive lineage trees and that's great. But really we want to quantify, right? We want to really measure some behavioral phenotypes of the cells. And so here I'm just sort of pointing out the kinds of things that we can extract from these lineage trees, including the types of divisions that the cells are making based on if a blue cell is giving rise to 2 blue cells, or 1 blue cell and 1 red cell, or 1 blue cell and 1 green cell, or if a blue cell is exhausting and giving rise to 1 red and 1 green cell. We can look at the cycling rate, so how long it takes for the cells to divide, and as well as motility, so distance, velocity, things like that. So to get back to that initial question that I had asked Diane at the beginning of my postdoc about why some MEP only give rise to one lineage or another, we started to look at the behavioral phenotypes of the disparate cells. So in the MEP, we generated lineage trees and collected motility data. We also did this for sorted megakaryocytic committed progenitors and erythroid committed progenitors. So these would sort of be like our ground truth for a meg committed and erythroid committed cells. And when we look at the lineage trees, we can see that the megakaryocytic committed progenitors have a much lower proliferative frequency. So they're only dividing a few times before those cells are actually maturing. And then we can actually visualize endomitosis, which is really cool to see. And so that's quite different than in the MEP where we have multiple rounds of division. And in the erythroid committed progenitors, we have a high amount of proliferation. We can also see that as those cells really start dividing, the rate of their divisions is much, much faster than those of the MEP. And then in terms of motility, I'm just sharing here some trajectory maps that we draw. Well, the algorithm draws them for us, that actually tracks individual cells. So each line here is a different cell. It's color coded with the same color code as the lineage tree. And right off the bat, you can appreciate that the MEP within the blue here have some motility, but the MKP, the daughter cells that are meg destined have a much higher motility. They travel much greater distances than the MEP does. And conversely, the daughter cells of the MEP that give rise to erythroid destined cells have very low motility. So you see them tightly cluster. And so that's also visualized in our sort of ground truth conditions, with the meg progenitors having motility and the erythroid progenitors really not. And so it worked out well that our cells really behaved differently depending on whether they were still bipotent or whether they had actually committed to one of the lineages. So then we went and compared time-lapse images of sorted MEP giving rise to all megakaryocytic colonies or sorted MEPs giving rise to all erythroid colonies. And this is really where we started to get information that gave us confidence to say that the cells that are giving rise, that the megakaryocytic committed cells are really much more motile and much more slowly dividing than a bipotent cell or an erythroid committed cell. And the erythroid committed cells are much less motile and much more proliferative and divide much faster. And so this was able to sort of quell my question about this idea that the reason we have MEP giving rise to all MK or all E colonies is not because of contaminating populations, but actually because there is a probability associated with lineage commitment in progenitors that have multiple potentials. So that was one vignette of how we used time-lapse microscopy. I have two others. I think I'm just gonna really quickly demonstrate this one point because this ended up being really important for my future studies. And that is, as I mentioned, having time-lapse imaging and lineage trees allows us to look at what cells do, what their functional output is. And so we had asked a question about what are thrombopoietin and erythropoietin doing at the level of the MEP. I will shortcut the story just to say that they do not instruct MEP lineage commitment. So it's not like you can feed a bunch of TPO to an MEP and expect it's going to make all MAGs, or vice versa, feed it a bunch of EPO and expect it to make a bunch of erythroid cells. In fact, these don't skew the ratio of functional output of the MEP. However, surprisingly, we did find, because we can look at the number of generations that an MEP persists and is able to give rise to 2 additional bipotent daughter cells as an expansion division, or 1 bipotent daughter cell and 1 lineage destined daughter cell, which we classify as a maintenance division, or an exhaustion division where an MEP divides and its 2 daughter cells are now restricted to 2 different lineages and there's no more MEP. What we found actually was that, so let me walk you through this because it's a little confusing. Looking along the control line here, so these are MEP in the presence of TPO and EPO. Here we're just tracking the number of expansion divisions over time in culture by generation. And what you can see is that the expansion divisions sort of peak around generation 3 and then slowly come down. And those are ultimately replaced by maintenance divisions, but those also slowly come down somewhere on the average of 7 to 8 generations. And then they all finally terminate in exhaustion. And when we follow this pattern and we look at MEP cultured in the absence of EPO or TPO, what we see is that the move from expansion divisions to maintenance divisions starts happening sooner. And so too do the exhaustion divisions. So what we're really being able to visualize here is the rate of exhaustion of these progenitor cells and their loss of bipotency. And so surprisingly, EPO, okay, I should say not surprisingly, TPO seems to be supporting that ability of the MEP to self-renew. And I say that's not surprising because TPO has already been demonstrated to play that role in HSCs and other hematopoietic progenitors. But it was not yet reported that erythropoietin could do the same. So now we have evidence to suggest that EPO is also promoting that self-renewal phenotype of the MEP. And the last vignette, I'll just very briefly explain. So this is some new data that we're generating in collaboration with one of my colleagues here at UConn, Joe Lorenzo, who's very interested in osteoclastogenesis. Osteoclasts are cells that resorb bone in our bodies. And these are actually mirroring osteoclast precursors that he's put in culture with the appropriate cytokines to promote osteoclastogenesis. And we're doing time lapse to actually watch the formation of these really huge, beautiful osteoclasts in a tissue culture plastic dish. But why I share this with you is because already, just in the few months that I've been collaborating on this project, we've already learned a ton that were falsely assumed in the literature. For example, Joe's hypothesis initially was that the velocity of the precursor cells would be very high, increasing the frequency with them sort of colliding with each other and fusing to start forming these osteoclasts. And I think you can appreciate in these movies, if I sort of play it back again, really the most motile cells in these movies, oh, I'm stuck. There we go. Really, the most motile cells in these movies are actually the larger osteoclasts. The precursors don't move quite as much. And so we're really excited to start to explore the field of osteoclastogenesis and also to try to help address this black box of, we know that osteoclasts come from the hematopoietic lineage. We know that they are in the macrophage lineage, but there's still that black box of how they actually commit to start fusing and forming these osteoclasts. So these are the things that we're hoping to address here. And I am totally out of time. So with that, I will just very generously thank all of the people in my postdoc lab, as well as Diane Krause, for introducing me to the world of time-lapse microscopy. And then my lab as well. It looks like I have a huge lab. Actually, I have an army of undergrads that do remote tracking. There's an entire conversation we could have about how we analyze time-lapse microscopy images, but it's still fairly labor intensive, and hence why we have so many students that work with us on this. And also I would very much like to thank our funding source, which is the NIDDK, that I have both a KO1 from. I've received previous P and F awards, as Diane was mentioning at the beginning of today's talk, and of course the University of Connecticut for giving me a startup fund with which to begin my independent research. Whew. I will stop talking now.
Thank you so much for your presentation. Questions? Anybody?
Hey, Shilpa's here.
Yep.
Oh, she disappeared.
She just bugged off.
Oh,
Oh,
there
there she
she
is.
No, I just turned my video on because I heard my name.
Hello.
Hello. How are
How
you?
are you?
Good. Nice to see you guys. Hi, Shangqin. Nice to see you too.
Hi. So what I did not touch upon is about the linear, the image annotation processing and making sense of it. That's a whole topic of another day, but I just, really this is on behalf of the Yale CCEH imaging course, what we want to give the field, provide some sort of assistance to lower that activation energy, to tell you it's possible, you can do this. We've all suffered through all sorts of strange questions to image them. So we would like to really share that knowledge and that sort of, gained through the trial and error, that hematopoietic cells, even though they're small, not adhering, they like to do all sorts of weird things, we still should be able to image them, as long as you have a sort of defined question, what you want to see.
It's true. And I'll selfishly plug, if anybody wants really in-depth training on this, I'm hiring postdocs. So call me.
That's not selfish. And if you want to advertise on the CCEH, send me a postdoc ad and we'll put it up there.
Excellent. Thank you.
Okay. Any other questions that anybody has? I actually had one about the imaging of the cells that were becoming an erythrocyte or a megakaryocyte. What was that tissue that you had so much color going on?
Oh, yeah. So basically what we're doing is isolating these primary cells from human bone marrow or mobilized peripheral blood. So we're sorting them out, and then we're plating them very sparsely in the semi-solid media.
So it's not like the whole shot out of a bone marrow. It's isolated specifically.
Yeah, this is all in vitro work so far.
It'd be fun to see if you could do that with a sort of slice of a chunk of the bone marrow.
Yeah, I'll tease and say I didn't have time to talk about it today, but we are doing a collaboration. So the group that I'm part of at UConn does a lot of skeletal work. So they're doing a lot of osteoarthritis models and fracture models. And although I didn't have time to talk about it today, we just started some 3D imaging, time-lapse imaging of fractured tibias, and watching the activation of the progenitor cells that are going to start forming the fracture callus for healing. And so those videos are blowing my mind, but I didn't have time to talk about it today.
You guys should make a connection with Ken and the Codex Core that's at Indiana, because they're actually working on having lots of markers so you can follow a lot of different cells. I think it's fixed tissue though. I don't think it's live cell imaging. So there are limitations to that, but it could be kind of fun to figure out if you could, pull that off together.
Yeah. Yeah, the group I'm part of here, this is the same group that made like the Cal 2.3 and Cal 3.6 mice. So they're really big into reporter mice, and they've crossed, I don't know how many colors.
That's great.
So that's the approach we're taking right now.
Yeah, that's a better approach, live. That's awesome. Anybody else? Want to thank everybody for attending, and just remind you to go visit our website so that you can find out about those pilot and feasibility funding opportunities. And I'm just going to share my screen again one more time real quick to tell you that the next,
Sorry, there's a question in the chat.
Okay, hang on. I'll come back to it. I just want to show you that the next imaging presentation date has been changed to March 20th. Ken Dunn's going to talk on light sheet microscopy. That was originally the 13th of March, and it's now shifted to the 20th. I missed the chat. Okay.
I think I'm gonna say it's Ching Ching. I'm sorry if I'm
Sure.
Go ahead and unmute yourself. One question. Do you think the cell numbers you seed in culture systems will affect their behavior?
yes.
Yes, I mean, it really depends on what you're trying to look for and how long we're talking about. Like any cells, even hematopoietic like progenitor cells, if you crowd them too much, they're going to be starved. They're going to be competing for that nutrients, and on top of that, it's the ROS generated during the light imaging or burning them. So these cell numbers you want to be sort of dense enough so that you can comfortably capture enough cells, but definitely do not you do not want to crowd them so that they behave like starved cells, crowded cells. Those needs to be titrated in the pilot, right? So before we can do that.
For sure. I call it the Goldilocks effect. You can't have too much and you can't have too few either. Because cellular crosstalk is a thing and it definitely helps support the cell behaviors. And so if you plate too sparsely, that also could potentially give an issue. But as Shangqin said, that's all things that you optimize as you're sort of building your approach.
Yeah, and also that we don't know this until you actually experience the one. If you plate too sparsely, the first thing you'll get frustrated with is you can't find your cells. So you want a comfortable number so that you know that you're looking at the right Z depths or the XY region. So we're talking about cells, not just gunk.
Yep.
Perfect. Anything else question wise, comments, help? Let me recommend that you reach out to the Yale Imaging Corps if you do have further questions. And want to thank you guys both for presenting today. Super fun to hear the technology that's new and informing on questions that we couldn't answer any other way. So it's really cool.
Thank
Thank
you
you so much.
Yeah. And thanks everybody for attending. And we'll end the meeting at this point if there are no other questions.
Finished. Yeah.
Okay, good. Thank you. Take care.